COVID-19 first emerged in Wuhan, China in 2019, where since then it has globally infected 359 million people and continues to claim the lives of individuals all over the world. Establishing herd immunity through vaccination is the only effective way of controlling the pandemic. The rollout of first-generation vaccines played a critical role in reducing viral transmission, hospitalization, and death. But with a virus like COVID-19, variants started popping up all over the world. We have seen the emergence of 5 new variants of concern since September of 2020. This posed a huge problem for our first-generation vaccines that were doing so well against the ancestral strain of COVID, and they quickly started showing signs of reduced effectiveness against the new variants. Doesn't it make sense to protect the tissue that's directly targeted by the virus in the first place? Absolutely, and this is exactly what a research team has investigated at McMaster University. They have designed a vaccine that is specific for building immunity within the respiratory tract by administering this vaccine intranasally instead of in the muscle. When designing these new vaccines, 2 vectors were compared, one coming from a human and the other coming from a chimpanzee. These vaccines contained 3 things: spike protein 1, a full-length nucleocapsid surface protein, and RNA-dependent RNA polymerase proteins. A mouse model was used to not only see the difference between the 2 routes of administration, but also the difference between vectors. After testing, what they found was that the intranasal vaccine gave a greater IgG antibody response against the spike protein compared to the intramuscular vaccine. They also noticed that the nasal vaccine gave systemic and mucosal antibody response, but the intramuscular vaccine only gave systemic coverage. They continued and found that the nasal vaccine gave sensitized B cells in the lungs, but again, the muscular vaccine did not. They then looked at how the vaccines might affect T cell responses and saw that the nasal vaccine was able to induce a significant number of killer T cells within the airway, but the muscular vaccine could not. The nasal vaccine didn't stop at B cells and T cells. They also saw that the nasal vaccine induced a training of airway macrophages, but no such training was seen in the muscular vaccine. When they compared the 2 different vectors, the chimpanzee adenoviral vector was superior in all categories discussed. This vector also protected against lethal infection by ancestral and new variants when compared to mice that were not vaccinated. Therefore, there was an importance in choice of route of administration, but also what vector to use to get the best protection. These findings suggest that a vaccine that is administered directly to the respiratory tract might be a better strategy to protect against an infection that targets the throat and lungs. Not to mention, the nasal vaccine was better at protecting against ancestral strains of COVID and new variants. Could this be the answer we're looking for? Could this be the future of vaccination for COVID-19? Since the study was done on mice and not humans, only time will tell what kind of change this new vaccine might provide.